Hello everyone. I welcome you to the week 2 live session of the NPTEL course Experimental Biochemistry, which is conducted by Professor Shagota Dasgupta and Professor Shomode from IIT Kharagpur. I am Bias Mukherjee, a research scholar in Dr. Shomode's lab from the Department of Bioscience and Biotechnology, IIT Kharagpur, and today I will be discussing with you some topics on amino acids and their corresponding, and then we will look into the corresponding questions. So is my slide visible to all of you? You can answer in the chat box or you can tell me by switching on your mic. Okay, so today we will be discussing some topics about amino acids, the pKa of amino acids, pi of amino acids, and then we will look into the different kinds of amino acids that are there, their biological role, and we will also look into the topics such as determination of the isoelectric point of an amino acid and determination of the isoelectric point of peptides. So I recommend you to watch the video till the end because this is going to help you a lot to answer questions in your competitive examinations like GATE, CSIR, NET, DBT and so on. So here we will be talking about very basic concepts first, and then we will be doing some of the related MCQs and MSQs. Alright. So this is a 2-hour session, live session, and you can ask me questions in between and I will be there to solve your doubts. So firstly, we will let's begin with the topics. So let us talk about amino acids. Now amino acids are the building blocks of proteins. So these are organic compounds that form peptide bonds together amongst themselves and they form these polypeptides. Polypeptides further associate to form the proteins. Okay, and proteins are the building blocks of life, as we all know. So amino acids are amphoteric in nature, which means that they can behave as an acid as well as base. So if you look at the structure of amino acids, so this is the structure of an amino acid, and you see that there is a COOH group, which is the carboxyl group, and this carboxyl group at low pH, what it does is it releases this hydrogen in the form of a proton. So it releases this H plus ion and it acquires negative charge. Again, so this is acting as a Bronsted-Lowry acid because it can donate H plus ion in a solution, right? Similarly, at a very high pH, what happens? This NH2 group, this accepts an H plus from the solution and it becomes NH3 plus, and therefore this is acting as a Bronsted-Lowry base. Therefore, we can say that amino acids are amphoteric in nature. Now, next, amino acids can be standard amino acids or the proteogenic amino acids. These standard or proteogenic amino acids are coded by genes and they are involved in the synthesis of proteins. So the standard amino acids are the common 20 amino acids that we know, and also selenocysteine and pyrrolysine. So pyrrolysine is found in bacteria and archaea, and selenocysteine is nothing but cysteine amino acid with a selenium group instead of sulfur. So we will be looking into the details of the structures and the properties of amino acids very soon. Now, besides the standard or the proteogenic amino acids, there are the non-standard or the non-proteogenic amino acids like gamma acetyl butyric acid, which is GABA. This is a neurotransmitter inside our brain. And then there is citrulline, ornithine. So all of these amino acids are the intermediates that are formed in the urea cycle. So like that, there are so many different non-natural amino acids. There is also this hydroxylysine and hydroxyproline that you study in collagen. So all of these amino acids, they are non-natural, because they are not involved in directly in the formation of proteins. All right, so amino acids are involved in essential life processes, and we will soon look into the details of that. So in brief, I can tell you that they are involved in building and repairing of tissues, enzyme activity, signal transmission, sensory perception, and so on. So looking into details into the amino acids that are produced in our body versus the amino acids that are not produced in our body, but again they are very essential for us, and therefore they need to be supplemented through our diets. So the amino acids that are produced in our body, they are called the non-essential amino acids. So they are produced in our body in sufficient quantity. Okay, so there are certain amino acids that are produced in our body but not in adequate amounts. So for that they need to be partially supplemented through our diet. These are arginine and histidine. Then there are the essential amino acids which are not at all produced by our body. So these are called the essential amino acids. They cannot be synthesized in our body, and the non-essential ones are synthesized by our body in sufficient measure. So here is a table that gives you the details of the essential and non-essential amino acids. So see, these essential amino acids, they cannot be synthesized from raw materials. They are 7 in number and they are obtained from dietary products. These all in all of these are essential amino acids. Arginine and histidine are semi-essential, and so see, these non-essential amino acids, they can be synthesized by the human body from the raw material, and therefore they need not be provided in our diet. They are 13 in number, but 1 in adults and 2 in children are slow to be formed. They need not be present in our diet. So these are all non-essential amino acids. Okay. So what is the biological role of amino acids? So if you look at the processes that happen in a human body, mostly majority of them are brought about by the amino acids, and that includes digestion and assimilation, that includes growth and repair of tissues, making hormones and brain chemicals, so all the sensory perceptions and neurochemical processes, all of these, then providing energy as a source, maintaining healthy skin, hair and nails, and then building of muscles, and then the entire immune activity, all of that. So again, sustaining a normal digestive system. So all of these processes are actively carried out by the amino acids in our body. So to understand exactly what is the role of every amino acids, let us look into this list. So these are the biological roles of the essential amino acids. So essential amino acids like phenylalanine helps in maintaining a healthy nervous system and boosting memory power. Valine acts as an important component in promoting muscle growth. Threonine helps in promoting the functions of the immune system. Tryptophan is involved in the production of vitamin B3 and serotonin hormones. So this serotonin hormone plays a vital role in maintaining our appetite, regulating sleep and boosting our moods. So very soon, now if you look into the functions of these amino acids, you can tell me what are the things that are going to happen to our body in absence of these amino acids. So if tryptophan is not provided in sufficient measure, of course our sleep is going to get affected, appetite is going to get affected, our mood is going to get affected as well. So very soon we will look into the diseases that are caused by deficiency of amino acid. So these functions are important because these can come in the form of match the following in your exams. So that is why I would recommend you to go through these, make a table, and then just go through the names of the amino acids and the functions they perform. So let's go about with these. Isoleucine plays a vital role in the formation of hemoglobin, stimulating the pancreas to synthesize insulin and transporting oxygen from lungs to various parts of the body. Methionine. Now, methionine is a sulfur-containing amino acid. This is used in the treatment of kidney stones, maintaining a healthy skin, and also controlling invasion. This should be invasion of pathogenic bacteria. Leucine is involved in promoting protein synthesis and forming the growth hormones. Lysine is necessary for promoting the formation of antibodies, hormones, enzymes, and in the development and fixation of calcium in bones. So you understand that if lysine is not provided in sufficient measure, there is going to be problems like weak bones such as osteoporosis. So now histidine is involved in many enzymatic processes, and histidine is a very important amino acid. We will learn about it very soon, because this is very important in maintaining charges of the proteins, charges of amino acids in proteins, and the charge of the consisting amino acids in a protein is very crucial, because that directs the functions of the protein in many cases. So charge of the active site of, say, an enzyme is going to tell us whether that enzyme is interested in binding to a ligand which is the opposite charge or not. So again, the synthesizing, so this histidine is involved in synthesis of RBC and WBC. Now we will look into the biological roles of the non-essential amino acids. So these are the amino acids that are produced inside our body, and so let's see what are the functions that they are involved in. So alanine functions by removing toxins from our body and in the production of glucose and other amino acids. And then cysteine acts as an antioxidant, and it provides resistance to our body. It is important for making collagen. It affects the texture and elasticity of skin. So glutamine promotes a healthy brain function, and it is necessary for the synthesis of nucleic acids, DNA and RNA. Glycine is helpful in maintaining the proper cell growth and its function, and it also plays a vital role in healing wounds. It acts as a neurotransmitter. Glutamic acids acts as a neurotransmitter, and it is mainly involved in the development and functioning of the human brain. Arginine is a basic amino acid, and it helps in promoting the synthesis of proteins and hormones, detoxification in the kidneys, healing wounds, and maintaining a healthy immune system. Tyrosine plays a vital role in the production of the thyroid hormones T3 and T4. So this is thyroxine 3 and thyroxine 4 in synthesizing a class of neurotransmitters, and melanin. Melanin is the pigment that provides pigmentation to skin, which are natural pigments found in our eyes, hair and skin. So serine is another amino acid which helps in promoting muscle growth and in the synthesis of the immune system proteins. Asparagine is mainly involved in the transportation of nitrogen into our body cells and formation of purines and pyrimidines for the synthesis of DNA, the development of the nervous system and improving our body stamina. So aspartic acid plays a major role in metabolism and in promoting the synthesis of other amino acids. Proline is mainly involved in repairing of tissues and in the formation of collagen, preventing the thickening and hardening of the walls of the arteries. That disease is called atherosclerosis. This is a very common term that you get in exams, and in the regeneration of new skin. So these are the different functions of essential amino acids and non-essential amino acids in deep details. Alright. So now there is a quick question for you. So based on your understanding of amino acids, what do you think about the amino acid supplements that people take? Okay, whether it is necessary or is it luxury? So what is your opinion on that? Well, I would say that we can get all the essential amino acids that our body requires through a balanced diet. Now, taking additional dietary supplements is a person's choice, okay? So again, now let us look at the deficiency of the amino acids. So since we understood how important the amino acids are for the normal functioning of the body, and how important they are in the life processes, we understand already that there are going to be a lot of different symptoms in the body because of the deficiency. So all of these complications include headache, weakness, irritability, fatigue, skin and hair related issues, insomnia, depression, loss of appetite, diarrhea and anemia. There are many more. You can search online and you can find a lot of different diseases that are associated with the deprivation of essential and non-essential amino acids in the diet of a person. Okay, now we will look into the properties of the amino acids based on the structure. So very recently we looked at the structure of the amino acids. Let us go back to that. Yes, so in the first slide, we looked at the structures of the amino acids. Now see, this is the acidic group, this is the basic group, this is a hydrogen, and then there is the R group. This R group is nothing but the side chain of the amino acid. This basic framework, all of these three groups, these are same in all the amino acids. But then this R group is different in each and every amino acids, and that is what gives unique identity to the amino acids. Alright, now let's go back to the slide where we were looking at the properties of the amino acids. So yes, so we'll have a look at the properties of the amino acids now. Let's go into slideshow mode. Yes. Okay, so based on the properties of the side chains, the amino acids are classified into various types. That includes the non-polar side chain. So the non-polar side chain is that this is mainly made up of carbon and hydrogen. So they are not going to be participating in intermolecular hydrogen bonding with their side chain. For example, alanine, glycine, alright, valine. These are all amino acids with non-polar side chain. If you look into this diagram, so these amino acids come under this, they are actually tiny. So glycine is a tiny amino acid because it has its R group is just hydrogen. Okay. Now instead of hydrogen, if you substitute that with a methyl group, it becomes alanine. So if you substitute the alanine with the OH group, it becomes serine. So all of these are very small amino acids. Now if you look at these are the aliphatic amino acids. Isoleucine, leucine, valine, these are the aliphatic amino acids because they are having just carbon and hydrogen in their side chain. So they mainly interact with the other non-polar side chain containing amino acids through hydrophobic interactions and side chain stacking interactions. Okay, so the polar uncharged side chain. So polar group means one of the atom is going to have partial negative and the other atom is going to have partial positive charge. For example, take serine. So serine has OH in its as its R group. Okay, so this O is a more electronegative atom and H is a more electropositive atom. So O is going to pull the electron pair towards itself, acquiring a delta negative charge, and H is going to have a delta positive charge. So this is a polar uncharged amino acid. So polar uncharged amino acids are involved in making intermolecular hydrogen bonding or ionic interactions. Okay, so these are involved in making hydrogen bonds with water as well. So they are non-ionizable, that means they cannot dissociate into H plus and O minus charges, but then they can be present and they can it can actually be involved in hydrogen bonding interactions. Now coming to the charged side chain. So amino acids such as lysine and arginine, they are positive, they are basic amino acids, so they have a positively charged NH3 plus in their R side chain, and amino acids such as aspartate and glutamate, they have an extra COO minus in their side chain. This renders them negatively charged even at neutral pH. Okay, so let us look at this figure. So all of these amino acids based on their R group, they are of different types. So there are aliphatic amino acids which include isoleucine, leucine, valine. There is a sulfur containing amino acids. So there are two amino acids that contain sulfur. One of them is methionine, the other one is cysteine. Okay, then is the aromatic amino acid. Aromatic group is like a benzene ring. It is having alternate single and double bond. Okay, so all of these amino acids, and that includes phenylalanine, tyrosine, tryptophan. You can refer to the names of the amino acids from here. So this is the single letter code, this is the name of the amino acid, and this is the three-letter code. Okay, for all the amino acids. And then so this is the aromatic amino acids, they are involved in all of these. Yeah, so that also comes under, some of them are coming under aliphatic interactions such as phenylalanine, and then there is tyrosine, there is tryptophan. Tryptophan is having an indole ring. Then there is histidine. Histidine may be charged, so it is coming in this circle. Okay, then all of these amino acids, they are involved in hydrophobic interactions. So all of these amino acids, methionine, phenylalanine, this is threonine. So hydroxylic, that is they are having polar uncharged side chain. So OH group is there in both serine and threonine, and that is why they are hydroxylic. They are forming hydrogen bond with water. Okay, and then there are the small amino acids, and that includes proline, glycine. Then all of these amino acids are the small amino acids. Then there are acidic amino acids. Acidic amino acids are asparagine and no, acidic amino acid mainly are these, aspartate and glutamate. So aspartate and glutamate, these are charged amino acid and they are negatively charged. Okay, so these N, they are actually containing nitrogen. Extra nitrogen is there in their structure. This is asparagine and glutamine, and then they are all polar amino acids. So 1 minute, I think someone is trying to enter. Yes. Okay, so we'll continue. Now this is the case with all the amino acids. Okay. Right. Now we move forward. So these are all the amino acids at a glance. Now it's very okay. 1 minute. You want to say something? Dr. Yogesh, do you want to say something? Please turn off the mic otherwise. I'm muting you. Please switch on the mic if you have to say anything, okay? I am not able to mute you. 1 minute. Yeah, so you've muted yourself. Okay, thank you. All right, please ask any question that you have. Okay, in the middle of the class or at the end of the class, whenever you're comfortable, please ask questions. You can type in the chat box. You can ask me directly. All right, I'll answer you. Okay, let's go back to this. Yes. So we will be looking at the amino acids now. Now this is glycine, there is alanine, there is valine. So glycine is having just this hydrogen as its R group. Now you substitute that with CH3, it becomes alanine. Now you substitute that with one CH2 and two CH3, so that becomes valine. This is how you can have a look at all of the structures of the amino acid. If you understand how exactly they are getting formed, it will be very easy for you to remember the structures, because it's actually important for you to remember the structures for the competitive exams. So see, if you substitute valine, if you just add one extra CH2 here, this becomes leucine. And then what you do is you take this CH3 and you put it here, then it becomes isoleucine, right? And then instead of this CH2, you add one sulfur here, this becomes methionine. Similarly, in case of phenylalanine, you just add a benzene ring. You add a benzene ring to alanine. It becomes phenyl and then alanine because this is a phenyl ring, right? And then there is proline. Proline is having this pyrrole group, which is a 5-membered ring. Okay, so the property of proline is that all of the amino acids are alpha amino acids, because if you look at this carbon, this is the COOH group, and this COOH group is connected to the C alpha carbon. To this C alpha carbon, this NH2 group is connected, and to this C alpha carbon, this R side chain is also connected. So in case of all the amino acids, this C alpha group is there, okay. But then in case of proline, what happens? This NH2 group is, it is not an alpha amino acid, rather this is a beta amino acid, because this NH2 group is somehow, it is connected to the beta carbon, and see, this is beta, this is gamma, and so this is how this is like, if you look into the structure of this is having the most unique structure compared to other amino acids. Now we move on to tryptophan. Tryptophan is having this indole ring. This is called the indole ring, okay. And then there is asparagine. So asparagine is having this NH2 CONH2 group. So CONH2 group is there in asparagine, and then there is glutamine. Glutamine is also having a CONH2 group, but there is an extra CH2 group here. And then there is serine. So in case of serine, what happens to alanine if you add an OH? So that becomes serine. And then if you add the OH here and you add a CH3 here, that becomes your threonine. So now all of these groups, all of these functional groups, I told you that they undergo protonation and they undergo deprotonation at different pH. So protonation means it is going to accept an H plus ion. Deprotonation is it is going to lose an H plus ion. So deprotonation is a property of acids and protonation is a property of bases according to Bronsted-Lowry concept, right? So now if you look into this, you see this COOH group, this actually loses this proton at a pH of 2.34, which is very, very acidic pH. So the gastric juices in our stomach, these contain HCl, and HCl there is present at a pH of around 2. So that is very, very acidic, and it is essential for digestion. It also kills the germs that enter along with the food. Alright. So if you look into this NH2, this NH2 accepts an H plus ion from the aqueous solution at a pH of 9.60. So all the amino acids, for all the amino acids, this COOH and this NH2 group is unique, right? So all of them will have values. These are called the pKa values. pKa means the negative log of acid dissociation constant. We have studied about this in the last class. So pKa is nothing but pH when the concentration of an acid is equal to the concentration of its conjugate base. That means that when this COOH group is giving out this proton and it is forming COO minus. So this COO minus is the conjugate base, and this COOH containing amino acid is the acid, right? So when the equimolar solution of this COO minus species and COOH species is there in a solution, that time what happens? It becomes log 1. So log 1, the value of log 1 is nothing but 0. And therefore, according to Henderson-Hasselbalch equation, this becomes pH equals to pKa. So therefore, we can say that this is having a pKa, the COOH group is having a pKa of 2.34 and this NH2 group is having a pKa of 9.60. Alright. So we can say that the pKa1 is 2.34 and pKa2 of this particular amino acid is 9.60. Then there comes pKa3. So who is having pKa3? Let us look at the amino acids with pKa3. So there are certain amino acids such as aspartate, glutamate, arginine. So all of these are having this ionizable side chain. Okay, so this is the ionizable side chain. Now this also, it is ionizable, that means it is going to get deprotonated at a particular pH which is 3.65. So this is the pKa of the R group and therefore it is called pKr. So that is true for all of these amino acids. Alright. Now we will look into this table. So if you want to have a look at the side chain of all the common amino acids and the pKa1, 2 and 3 of all the common amino acids, you can get an idea from this chart. You can look at the pi. Pi is nothing but the isoelectric pH. So this is a pH at which the pKa and pKa1 and it comes between the pKa of these respective chemical groups such that there is a population of species that is having net neutral charge. So we will look into that very soon, and that is called the pi of the amino acid. So at the pi or the pH at which the amino acid is having net neutral charge, this protein actually precipitates. All right. So this is a very important concept in biology. So when we make buffers, as we looked into buffers in our previous class, we learned that the pH of the buffer should be 1 unit plus minus the pi of the pi of the amino acids to make sure that they are not getting precipitated. Okay, so you can have a look at all the different properties of the different amino acids, their pi values, and we will very soon learn to calculate the pi of the amino acids. Okay. So I am sure you have been bored by now with so much of theory. So let us start with the questions. So we have brushed up the concepts. Now we will look into the questions little bit, and then we will go into other concepts again when we have to solve those questions. Okay. So these questions are with respect to whatever you have learned up till now. Alright. So this is week 0 assignment. Let us look at the questions. So the first question asks you, what is the one letter code for the tetrapeptide? Tryptophan, asparagine, tyrosine and lysine. So we just learned the single letter codes. So what do you think? What will be the right answer? You can tell me or you can write down in the chat box. I give you 1 minute for this. It's a very simple question. Let's see if there are any answers. So are there any answers? There are no answers, no problem. We'll discuss. Come on. Okay. So let us look. So tryptophan is going to have a single letter code W. Okay. Asparagine, this is going to have a single letter code N. Tyrosine is going to have a single letter code Y, and lysine has a single letter code K. So let's see if so according to me the right answer is C. Let us check if this is correct. Okay, that is correct. Alright, now let us look at another question which asks you about your knowledge of the side chain of amino acids. Okay. Now that we have studied that amino acids are having different properties and the properties are mainly based on the side chains of the amino acids, let us look into this question. So they are asking you the examples of amino acids with basic side chains. Basic means there are going to have more than one basic groups, and basic group is NH2. So we understand that there are going to be more than one basic group so that it can have a non-neutral charge at pH 7. So which one of these amino acids is going to be basic at neutral pH. You have 1 minute to solve this question. All of these are very simple questions. Try out. Yes. Okay. C. Okay, you say C. Let's find out. So C is alanine and glycine. Is that so? Let's see. Okay, let's see the right answer. Okay. So if we look into the right answer, well, it is not C. It is lysine and arginine. So that is your D. Now, why exactly is that so? So they are having basic side chains. That means if you look at the structures of this amino acid, lysine and arginine, so see, this is the COOH group. Now this is having a pKa of 2.2, and then there is the alpha amino group, which is again having pKa of 9.0, and then there is a epsilon amino group. This is lysine, right? So there is an epsilon amino group, and it is going to have the pKa of 10.5. Okay, now this is arginine. Arginine having pKa of carboxylic group, it is 2.2. pKa of alpha amino group is 9.0, and the pKa of this is called the guanidinium group, guanido group. Okay, so this is having a pKa of 12.5. Now let's see at pH 1. Okay, this is a very acidic pH. So at pH 1, what happens? This deprotonation has not happened. So this is present in the form of COOH. But then these are there, so they have not been deprotonated yet, right? So they are present in the form of NH3 plus, and this is present in the form of NH3 plus. So therefore what happens? The pH is 2. Okay, and then what happens? As you slowly add NaOH, you titrate it with a base. What happens? pH 3 is obtained. Sorry, yeah. So in pH 3, what happens? The net charge is +1. Why? Because in pH 3, this COOH group has undergone deprotonation. So then what happens? It is having a negative charge. All right. Then again at pH 7, it is having again +1 charge, because see, all of these values are more than pH 7. So then at pH 10, what happens? This NH3 plus, it loses it loses one H plus, and it becomes NH2, and therefore at pH 10 it is becoming negatively charged. Again at pH 11, what is happening? See, this NH3 plus, its pKa is 10.5, right? So at pH 11, it is losing this H plus ion, it is becoming NH2. So then this is uncharged, only this group is charged, and so that is -1. The charge is -1. Okay. Similarly, for arginine, if you look at it, these are the different charges. So for arginine, this is 12.5. That means even at pH 11, it is going to be positively charged. This is going to have no charge. This is going to have negative charge. So that is 0 charge, right? And then at pH 1, what is going to happen? This is going to have no charge, this is going to have no charge, and this is going to have negative charge. So that becomes -1. And therefore we can say that at neutral pH, this is the neutral pH, which is 7. So at neutral pH, these two amino acids, they are going to have positive charge. So they are going to have basic side chains. Is that clear to you? Okay, you understood? We'll go to the next question now. All right. Okay. Right. So now this is week 2 assignment. Okay. So they are asking you the pair of amino acids containing aromatic side chains. So these are the different amino acids. Tell me, it's very simple, which one has aromatic side chain. Aromatic means it is going to have a benzene ring kind of side chain. Okay, so look at the structures of the different amino acids. This is phenylalanine, this is tyrosine, this is histidine, and that is tryptophan. These are all the amino acids that contain these alternate double bond and alternate single bond structure. This is called conjugate double bond structure. Okay, so these are the amino acids that are actually aromatic in nature. So amongst these, which two do you find here? Yes, that is the first option, or phenylalanine and tyrosine. Now we will look at another question again on the side chain of amino acids. Now these are actually very simple questions. Okay, but then they are very scoring. For these, once you go through the videos of Professor Shomode and Professor Shagota Dasgupta, these will become very simple for you. But then they are very scoring. So it's very important that you practice both the easy types and the difficult ones. We will come to the difficult ones very soon. Okay, so stay tuned. Now, which of the following pairs have imidazole and indole ring in their structure? So imidazole and indole ring, these are present in two amino acids. Let us look into them. So this is histidine. Histidine is having this 5-membered ring. This is known as the imidazole ring. Now this is very important in protein purification using the nickel NTA chromatographic column. Okay, so this indole ring actually has affinity for nickel and it binds to nickel. That is why this nitrogen, this actually donates electron pairs and it forms a conjugate bond and it forms a complex with nickel. Okay, and so this is very important in protein purification. So we make this, we add histidines externally to nickel and we make this histidine tag, hexahistidine tag. So we will learn about all of these in the upcoming lectures. So this is indole ring. Now indole ring is found in tryptophan. Indole ring is very important because this gives you a lot of absorbance, and therefore tryptophan is a very important amino acid when it comes to quantification of proteins, because this absorbs a lot of light in the UV visible spectrum. Okay, so the right answer is histidine and tryptophan respectively. Now we will look into the stereochemistry of amino acids. Okay, so first is the L and the D form. So a lot of people get confused between this capital L, capital D and the small l, small d. So we will look into this and we will clear your doubts once and for all. Okay, so first is, what is chirality? Chiral carbon is a carbon to which 4 different chemical groups are attached. Okay, so if you look at this alanine, this is the carbon. So to this NH2 group is attached, COOH group is attached, hydrogen is attached, and CH3 group is attached. Alright, so these are 4 different groups. Because of these 4 different groups, the amino acid is chiral. Now if you look at glycine, it is having one COOH group, one NH2 group, but then it is having 2 hydrogen groups. So this makes it achiral. So glycine is the only amino acid that is achiral in nature. Okay, so we'll go back to the slide. Yes, so glycine is the only amino acid that is achiral in nature. Baki all amino acids, they are chiral. Okay, some amino acids even have more than one chiral centers. We will learn about them as well. So now next thing is, what is D and L form of amino acids? Now if you look at your hands, okay, so look at your left hand, you see that this NH2 group, this is pointing towards your wrist, towards the inner side. Okay, if you place both of your hands like this NH2 group is actually pointing towards the inner side in Fischer projection formula. If you look at this amino acid, the NH2 group is pointing towards this left inward direction, right? So this is called L amino acid. So L amino acid is when this NH2 group is towards the left, and in the Fisher projection formula this is actually pointing towards the outward and towards the inside of your um, body, okay, towards your body. And then this is the D-alanine. In D-alanine, what happens? The NH2 group is present towards the right. So this is the capital L and capital D forms of amino acids. So in nature, mostly we find L amino acids. But then that is not all. We also find D amino acids. We will soon find out exactly where. Okay. So the D amino acids are just this L and D amino acids. These are basically mirror images of one another. Now if you look at dextrorotatory and levorotatory, let us find out what they are. So this is a light source, okay. Now in normal light what happens? The electric field and the magnetic field vectors are all perpendicular to one another and they are constantly vibrating. But then there is a polarizer. So what polarizer does is it is like a slit, okay. It allows only light in which the electric field vector is vibrating in one particular direction to pass through. Now what happens? This is called the plane polarized light. That is, the electric field vector is perpendicular to the plane. That means if the slit is like this, okay, this is a window, and through that window only the light whose vector is rotating like this up and down, only that passes. Baki, whatever is rotating this way, whatever is rotating in sideways, all of that is removed. All of that is blocked by this window. This is the polarizer. Now this plane polarized light, when it comes to this amino acid sample, now this, because of the chirality of the amino acids, they can bend this plane polarized light. Okay, this is a very interesting property. They can bend this plane polarized light towards the right or towards the left. Now, here they have bent it towards the right. If they are bending this plane polarized light towards the right, it is called a dextrorotatory compound. Dextrorotatory is the small d. Small d form, this is dextrorotatory, and it rotates plane polarized light towards the right. Now there is the L form. L form is levorotatory, and this rotates plane polarized light towards the left. Now when L and D forms are present in equal measure in a mixture, then it is known as a equimolar mixture or racemic mixture. Then what happens? Some of the molecules rotate this plane polarized light towards the right, the others rotate it towards the left, and net rotation is 0. Okay, that is the property of racemic mixtures. So amino acids, the amino acids that are present in our body, they are mostly this capital L amino acids. Okay, now looking at D amino acids. Now D amino acids are present in nature. So if you look at this structure, this is actually the peptidoglycan layer found in the cell wall of bacteria. Okay, so these bacteria, what happens is they are having N-acetyl glucosamine and N-acetyl muramic acid. This is NAM and this is NAG. So this is to this NAM, these 5 amino acids are connected, and these are actually cross links. These are basically bridges. So there is a layer of NAG and NAM, and again a layer of NAG and NAM, and these are connected by these bridges. They are the cross bridges, which is made up of these 5 amino acids. This is a pentapeptide, and they are made up of alternating D and L amino acids. See, this is L-alanine, the next one is D-glutamic acid, then again L-lysine, and then there is D-alanine. So this is how these are present in bacteria. Okay, now we will look at the questions on stereochemistry of amino acids. Now, which amino acid do you think has the most number of chiral centers? Let us look at the structures of the amino acid, because without the structures it is very difficult to understand. Alright, so we will be looking at the structures of the amino acids. So based on the structures, which amino acid do you think has the most number of chiral carbons? So I can already see 2 here. I can see 1 here. How many do you see here? Right? I see 2 here. What about this one? No, I don't see many. Right? Let us look at the answer. So the answer is 1 and 4. 1 is isoleucine. Isoleucine, if you look at this carbon, this is having one CH3 group attached. This is having this entire big group attached to it. This is having this hydrogen attached, and it is having this ethyl group attached. Right? So then this is a chiral center, and this carbon, this C alpha carbon, I told you this is always a chiral center. Why? Because it is having NH3H COOH group, and it is having this R side chain. So all the 4 are different groups, and therefore this is a chiral center and this is a chiral center. Similarly for threonine, if you look into this is definitely one chiral center. Here again 1, 2, 3 and 4. There are 4 different chemical groups, and therefore this is also a chiral center. So we see 2 chiral centers here. So what about serine? This is having only one chiral center. Why? Because this is definitely a chiral center, but here you see 2 hydrogens. So they are same chemical groups attached to the same carbon, and therefore it is having only one chiral center. So this is not our answer. Here again, if you look, this is CH2. So definitely not a chiral center. Here if you look, it is having similar kind of groups attached, C and H everywhere, right? So this is also not having chiral centers. Right. Now we look into the isoelectric point of amino acids. So I told you before, isoelectric point refers to the pH at which the net charge on an amino acid is 0. Okay. So initially at very high acidic pH, what happens? There is completely this NH3 is existing as NH3 plus. The COOH group is existing as COOH. Now what happens at intermediate pH? Let's say neutral pH, okay? This COO minus, this has lost one H plus, so it is getting deprotonated, and this NH2 is going to be present in the form of NH3 plus still, because this is having a low pKa value and this is having a high pKa value. Its pKa value lies in the range of pH 9, and this one's pKa value lies in the range of pH 2 point something, right? So then what happens, as you increase the pH, this also undergoes deprotonation. It donates this H plus and it assumes neutral charge. So then what happens? This is having negative charge, this is having 0 charge, so it is minus 1. Here it was plus 1, 0, so that is plus 1. But here what happens? Both of them are having charges. This is having minus 1, this is having plus 1. It gets cancelled out and you get net 0 charge. Alright. So a typical amino acid with neutral R group transitions from positively charged to a zwitterion. This is known as the zwitterion. So zwitterion is a chemical species which is having both positive and negative charges on it, and they are equal number of positive and equal number of negative charges, okay? And then to a negatively charged moiety, which as the pH increases. So as the pH increases, this becomes negatively charged. Now, how is a zwitterion formed? So zwitterion is basically nothing but a salt, okay? That is formed because of acid-base reactions. So what are the acid-base reactions? This is what an acid is donating H plus ion. Here what is happening? Over here, this base is actually this is initially it had taken an H plus ion and then it is giving away that H plus ion, okay? So what happens? Deprotonation or loss of H plus of COOH group as pH increases, and if you go this way, this NH2 actually takes up this H plus ion and it becomes positively charged. So that is protonation of the NH2 group as the pH decreases to give NH3 plus. This is how zwitterions are formed. Okay. Now again our famous equation, you will get this in every exam. This is Henderson-Hasselbalch equation. So this actually equates pKa with pH, and it tells you about the concentration of the acid and the conjugate base in a solution. Similarly, this can be drawn for this is actually done for a weak acid. Similarly, it can be done for a weak base. and this is the measure of this pKa is nothing but a measure of the acidity or basicity of a solution. Okay, so what are they telling you? That according to the Henderson-Hasselbalch equation, pH is equal to pKa when the HA, this is the acid. So the amount of acid present in the solution is equal to the amount of the conjugate base present in the solution. That is, the amount of COOH present is equal to the amount of COO minus present. That means equal amount of this moiety has broken, equal amount of this compound has broken to give this compound, right? So these two species, they are at an equilibrium. That is when this becomes log 1, because this and this cancels out, the concentrations are the same. So this becomes log 1, the value of log 1 is 0, and therefore pH is equal to pKa. Alright. So now lower pKa means that the acid is strong, okay, and higher pKa means that the acid is weak. That means at lower pKa what happens, even if very less amount of H plus is actually dissociated, easily the acid, the same amount of conjugate ions are formed, and then it becomes like it actually gives away this COO. H gives away H plus ion. So lower pKa means that the acid is strong. So all of these has been discussed in details in our previous video. So pKa is the pH at the midpoint of the flat buffering zones in a titration plot. So if you look at the videos, if you look at the lectures, then Professor Dasgupta and Professor Dey has taught you about the titration curve, then you can look at the buffering zone. So here if you look, this is the positively charged species. So at lower pH, this positively charged species dominates. At neutral pH, Zwitter ions dominate, and at higher pH, the negatively charged species dominates. Exactly what we saw here. At lower pH, this is having plus charge. At higher pH, this is having minus charge. And at intermediate pH, this is having charge 0. So this is the point where Pi is found, and these A and B points are where the concentration of the acid is the same as that of the conjugate base. Okay. So how to calculate the isoelectric point of an amino acid? So if you look at this structure of the amino acid, so you have the pKa, this is of the alpha COOH group, and then you have the pKa of this R group. This is the pKr, okay, this is the R group, which is COOH, and then you have this NH3 plus. So now how you do it is, you have to find out exactly at what pH the net charge is going to be equal to 0. So at pH 2, what is going to happen? This is going to become negatively charged because this H plus is going to be lost. So this is okay. So this is going to have plus charge. This is going to have 0 charge. No, sorry. This is going to have plus charge. This is going to have negative charge, and this is going to have 0 charge, right? Because this is still COOH at pH 2, and so therefore you take this value and you take this value, because what happens at pH say 4 or pH 5, this OH is lost and then this becomes COO minus, and so the two pKa values closest to the pH at which the net charge is 0, you take those two pKa values and then you find out the Pi. Alright, so this is the acidic amino acid and aspartic acid, and the Pi is calculated as the two pKa values divided by 2. So for neutral amino acid, this is very simple. You just take pKa1, you take pKa2, and then you find out the average of them, and then you get the neutral amino acid Pi, which is 6.01 in this case. And then you, for the basic amino acids again, you see this is not very important here, because what is happening at pH 3, this is becoming negative charge, but then these two are having plus charge and plus charge, so that is plus 2 initially, and then this becomes plus 1. right? Because this actually becomes -1 at pH 3. And so this is +1 and -1. Then at pH 9, what happens? This actually loses H plus ion and it becomes NH2. So the net charge now is 0, -1, +1. So the net charge is 0 here. Okay. And again here what happens? At pH 11, this is lost and it becomes 0. So the net charge is -1. Therefore, the two pKa values that are closest to the pH at which net zero charge is found are these and this. So therefore, you take these two values and you find out the pi. Alright. So pi of a diprotic acid or neutral amino acid is calculated using pKa 1 and 2. To find the pi of a triprotic acid, you need to take the average of the pKa values that are closest to the pH at which the amino acid assumes net charge 0. That is used. Okay. So this is all. Now we will look at the questions on pi. Okay. So you can take a 5-minute break and then you can tell me the answer of the question, or you can write in the chat box. Okay, whatever is comfortable. Let us take a break for 5 minutes and then I will be back soon. Okay, let us begin again. We'll resume. All right. So now these are the two questions. Sorry. Okay. So all right. Let's look at the questions. Yeah. Which of the following statements is or are true for isoelectric point? Can you hear me? I'm audible, right? Okay. All right. We'll look into the questions. Yeah. Okay. So the questions on PI are, which of the following is true regarding isoelectric point? So they are asking you, what is exactly isoelectric point? So the options are the pH where the molecule carries no net electric charge. The second one is the pH where the carboxylic group is uncharged. And then there is the pH of the maximum electrophoretic mobility. And then there is field point. Okay. Now, according to the definition that we know, which one is it? What do you think? You can answer in the chat box. You can unmute and tell me, or I can tell you the answer. Okay. So, based on whatever we studied, it is this one, because it is the pH at which the molecule carries no net electric charge. Is it the pH at which carboxylic group is uncharged? No, because carboxylic group is uncharged at a very, very, very acidic pH, right? That cannot be the PI. All right. And so PI, the PI has maximum mobility. No. What happens is, this is a nice thing. So what happens when the molecule is having no net charge and you apply an electric field, what happens? The molecule does not move at all. All right, that is a very interesting concept. So all of this electrophoresis process happens when the molecule is actually negatively charged, be it DNA, be it RNA, be it proteins. All these agarose-gel electrophoresis, SDS page, all of these in all of these processes, the molecule is negatively charged and then it moves from the cathode to the anode. Okay. So that is how the anion is going to the anode. But what happens if the molecule is uncharged? It will simply not move in an electric field, and so therefore the right answer is the pH at which the molecule carries no net charge. Next is, which of these amino acids have PI close to their physiological pH? So I told you this answer while teaching you about the different amino acids. Let's try to remember. Okay. So if you look at this structure, this is actually the structure of histidine. So yes, this tells us that the first answer is definitely correct. Now this is the structure of histidine and this is the imidazole ring. Okay. So what happens at pKa of 1.8. Now this is a very, very low pKa, very acidic pKa. What happens? This COO minus group, this loses this H plus ion, it becomes negatively charged. Then at pKa of around 6, this NH3 group, this loses H plus ion and it becomes NH2. Oh sorry, this side chain NH2, right? So this side chain NH2, it loses H plus ion, it becomes NH. And then what happens at a very high pKa? This alpha NH3 plus group, this loses H plus ion and it becomes NH2. So therefore, the right answer is histidine, because physiological pH ranges from around 6.4 to 7.4. Alright. So it is very close to pH 7 or the neutral pH. That is the physiological pH. And therefore, histidine is a very, very important amino acid when it comes to enzyme activity and all of different life processes. Okay. So the next question is again on the charge of amino acid. Let's find out what is there in this question. So when amino acid alanine is having R group CH3, right? It is added to a solution with a pH of 7.3. Alanine becomes, they are asking you, okay, what charge it is going to have. Okay. So alanine is basically an amino acid which is having pKa pKa1, that is the pKa of COOH, it is in the range of 2, and the pKa of its NH2 group, this is in the range of 9.6 or something, right? So this is 2 and this is 9.6. So you add both of these up, it becomes 11.6, and then you divide that. So that becomes what? 5 point something, right? So now what happens? They are asking you in the solution of pH 7.3. So from this 5, pH 5 or 6, let's take 6, okay? So pH 6 to pH 9, it is going to have 0 charge, and therefore it is going to be a zwitterion. That is in my opinion. Let's find out the right answer. Right, so see, its pKa 1 is 2.34, pKa 2 is 9.69. You add that up, you divide that by 2, it should be by 2. Okay, it's not given here, it's not mentioned here, but we have already learned that this should be by 2, and so the isoelectric point lies around 6, and therefore the right answer is definitely a zwitterion, because from pH 6 to pH 9.69, it is going to be a neutral charge, but both positive and negative moieties are going to be there, and therefore it's going to be a zwitterion. Right. So now comes another question. This is very interesting. So they are asking you to calculate the PI. Now these are the type of questions that you are definitely going to get for all the competitive exams. So you should be very well versed with these questions, and these are the easy ones. We have still not stepped into the realm of difficult questions yet. Okay. So they are asking you, this is the pKa1 of COO minus, this is the pKa2 which is of this alpha amino acid, and then comes the pKar. So this is definitely lysine, because this is having this epsilon amino group, right? So they have given you all the values, and they are asking you what is going to be its PI. So what do you think is going to be its PI? Well, this group initially it is HCOOH, right? So it is having plus, that is 2 positive charges. It is going to be plus 2 initially. Then what happens at pH of say 3? This group is going to get group is going to become COO minus. So that becomes total net charge is now plus 2 minus 1. So that is equals to minus 1, right? It becomes minus 1. Then what happens at a pH of 9.6? This guy is going to become NH2. So the entire molecule is going to become 0. Alright. So then this is an important pH. And then what happens here? At the pH of 10.54, this guy is going to become NH2. It is going to become uncharged. So the molecule is going to become what? Negatively charged, right? So we will take the values that are closest to the pH at which the net charge is going to be 0. So which two values are we going to take? We are going to take this value and this value. Okay. So then we calculate using PI, and then the answer is definitely 9.8, because this is 9.06 and then 10.54. You add that, divide by 2, you get 9.6. Right? So all the pKa values are given here. Now we go to an MSQ. Now these MSQs are very favorite questions of the exam setters, the paper setters of GATE examination. Okay, so they are going to ask you a lot of MSQs. CSI and NET exam also they are going to ask a lot of MSQs. Okay, so you should be very well versed with these MSQs. You must practice a lot so that you can be very used to MSQs. Okay, so this is on the nature of amino acids. It's a simple question. Let's look at it. So which of the following is or are a set of polar amino acids? So which of these are polar amino acids? Tell me. Aspartate, asparagine, threonine. Serine is a polar amino acid. Threonine is also a polar amino acid. Aspartic acid is a charged amino acid. Lysine is also charged. Phenylalanine is not polar, right? It's just a benzene ring. Tryptophan, it is not polar. Alanine, not polar. So that means this gets cancelled. Methionine, not polar. Proline is there, lysine is there. Proline is also not polar, right? So then it should be C and D in my opinion. Let's look at the answer. So here they are giving you the structures of all the different amino acids. Look at asparagine. It is having this CO group, and then it is having this NH2 group. So see, this is definitely polar. It can be a polar amino acid, right? So this threonine also, it is having this OH group, which is a polar group. It can be a polar amino acid. So there is threonine, there is asparagine, and then there is aspartic acid. This is obviously a charged and polar group. And then there is serine. So these two options, in my opinion, should be right. Yes, they are right. Okay. Now we will go into a question that deals with titration, pH titration of amino acids. Now, how did people suddenly come up with this pKa values? Well, people actually did a lot of titration experiments. So in titration experiments, what you do is you take an amino acid solution, and then to that you first add a lot of acid. You take the solution down to a pH of, let's say, 2, and then you gradually add, drop by drop, you add this 1 molar solution of an alkali, say NaOH. Okay, then what happens, as you keep adding, the charts, the pH curve that we have studied for glycine, for different amino acids, that appears, right? So what happens, as you keep on adding this NH2, as you keep on adding the bases, the initially what happens, this COOH, this gets converted into COO minus. So initially there is a rise, and then there is a straight line. Okay, it becomes stagnant, and then it increases rapidly, and then again there is a straight line. So the point where it increases, and this point is exactly where this pKa2 comes into play, and this deprotonation of this NH2 group happens. And in the region where there is these straight lines, these are the buffering regions. So these are the buffering zones, and at the middle of the two buffering zone is where the pi lies. All right, so they are asking you that they have given you the pKa and the pKa1 and pKa2 of an amino acid. They are asking you its pi. Now I have not included the graph over here because you can calculate pi just using these two values, and the graphs and all you can find from the NPTEL course videos. All right, so this is the answer. You can calculate pi very easily by taking these two values and getting an average of these two values. Now let us look at this question. During the pH metric titration of glycine with NaOH, I told you the entire process. Now let us look at the question. Okay, certain amount of HCl is added at the beginning. I just told you the answer. I hadn't noticed that the question was asking you the same thing. Let's see if you can answer. The role of HCl is to neutralize NaOH, neutralize glycine, completely protonate glycine, convert glycine to zwitterionic form. So which one do you think would be the right answer? It cannot be neutralize NaOH because HCl is added long before we are adding NaOH. So definitely it won't travel through time and neutralize NaOH. Okay, it cannot neutralize glycine because glycine is initially having positive charge, but then when you add HCl, it is not really going to neutralize glycine. It is not going to form a compound with this Cl minus ion. Okay, it is completely going to protonate glycine. Well, initially at a very medium pH, it is going to be a zwitterion, right? Glycine is going to be a zwitterion. Then what happens when you add more H plus, glycine, the COOH is going to actually take up, the COO minus is going to take up this H plus ion at its respective pKa1, and it is going to be protonated. It is going to give you positive charge. So exactly we are going to begin with positive charge, and we are going to progress to net neutral charge. Then we are going to go to negative charge for the amino acid in pH metric titration. And therefore The right answer should be completely protonate glycine. It converts glycine to zwitterionic form. That is not its role. That is going to happen only when we begin from a pH of, let's say, 12. So we are going from pH 12 to medium pH, then in that case HCl would be added. But they have already asked you that they are adding it at the beginning. So that means from zwitterionic they are going to low pH, right? So the answer is given here. Yes, it's completely protonate glycine, and in this case it was just simple PI calculation using the pKa1 and pKa2 values. Okay, then we will look at this question regarding the pH metric titration of lysine. Now if you look at this curve, this is the initial structure of lysine at very low pH. See, it's pH around 0 point something. Okay, so that is very acidic pH. Then what happens? pKa1 is reached. In pKa1 what happens is this OH group gives away, when a COOH group gives away H plus and it becomes COO minus. So here is what pKa has been attained. Then what happens? There is this very sharp rise, and then there is pKa2. Now what happens? Here the equivalence of OH, they have mentioned it in the form of equivalence of OH. Now here actually lies your neutral pH, alright. See, this region is the neutral pH. So that means here the neutral pH is going to lie, and then comes what? Then comes this the R group that is going to get protonated, and then there is this H, the negatively charged alpha amino group that is going to get protonated. So then here what happens? It was having initially +2 charge. Now it is having 2 plus and 1 minus. So that is +1 charge. Here it is having what? 0 and plus. Minus and plus. So that is net 0. So this is the region where Pi is going to lie. And two pKa values next to Pi are pKa2 and pKa3. So you take these two values and you calculate the Pi, right? But then in the question they have not exactly asked you Pi. So what are they asking you? When NaOH is added to a solution of lysine dropwise, the number of inflection points, that is sharp changes of Pi, sharp changes in pH are obtained. Okay, so how many inflection points are there? So if you look into this, this is definitely one of the inflection points, that is very sharp change in Pi. Okay, so the answer should be 1, because there is only one inflection point that is there. So next is question on pKa, pKa of amino acids. So this question they ask you the pKa2, that is the pKa for the side chain of histidine is 6.0 when it is present in an isolated state. Now here actually they are asking you about the difference in the properties of an amino acid when it is present in the free state versus when it is present inside a protein. You can see inside a protein there are a lot of amino acids. All of them are projecting their own characteristics, right? So the environment also affects the properties of an amino acid. Now this question is actually on that. So considering a specific protein with histidine at its active site, yes. So the homework for you is to find out which all proteins have amino acids, which all enzymes have histidine at their active site. Now this would be very interesting if you can just look up and find out, because there are a lot of different enzymes that have histidine at their active site. So the pKa2 is expected to be less than 6 if the active site has more positive charges. Less than 6 if the active site has more, okay, greater than 6 if the active site has more positive charges, less than 6 if the active site has more negative charges, same like isolated histidine, that is 6. Well, this cannot be the right answer because the properties of a protein are influenced by its environment. Okay, so what happens is less than 6 if it is having more positive charges. Greater than 6 if the active site has more positive charges, or less than 6 if it is having more negative charges. I give you 2 minutes to find the answer out and to tell me, okay? Take some time because this is a very interesting question. Okay, so let us look at the answer. Well, you have already looked at the charges of histidine. So this is the side chain. So now what happens in this case is that when histidine residue is present in an active site having mostly positive charges, the pKa becomes lower than 6 because positive charges in the active site destabilize the positively charged histidine side chain. Okay, so histidine side chain was positively charged. It should have become uncharged at pKa 2, which is 6. But then what is happening? It is destabilizing. All of these positive charges destabilizes it, and so a higher concentration of protons, lower pH is required to protonate this histidine side chain, therefore resulting in lower pKa. Alright, so it is going to be less than 6 if it is having more positive charges. Now comes a very interesting part, which is how to determine the pi of peptides. So now we have looked at the concept that these amino acids, they are the properties of the amino acids, the charges of the amino acid. This is all influenced by the environment of the amino acids, right? So now let us not look into the concept of proteins. Proteins are having like a lot of amino acids, hundreds of amino acids. We would just look at a tripeptide. So tripeptide is having 3 amino acids linked together, and let us find out how the presence of these 3 amino acids can influence the pi of this entire tripeptide. Okay. So firstly, this is a tripeptide made up of aspartate, glycine and lysine. So first and foremost what you do is you look at all the ionizable groups. Okay. So what are the ionizable groups? Now this is the C-terminal group, which is pKr. It is having a pKr. No, 1 minute. Yeah, so this is the N-terminal residue. This is the N-terminal residue, this is the middle residue, and this is the C-terminal, okay? So this N-terminal residue is actually having a side chain, which is COOH, and this is having a pKr of 3.65. Alright, then there is this NH2 group, which is having pKa2 of 9.6. Now this is already a peptide bond, it is not ionizable. Here also not ionizable, this is glycine. Then what comes is lysine. Lysine is going to have this R side chain, which is ionizable. It is 10.53, and then this is the COO minus group, which is ionizable, and it is having a pKa of 2.18. Now, how do you determine the pi of peptides? Well, what you do is, initially, what is the charge of the peptide? This is definitely going to be present as NH3+. This is going to be present as NH3+. So initially there were +2, charges are +2. Then what happens? At this pi, at this pH, this guy becomes COO minus. So this becomes -1 charge. Then what happens at a pH of around 4, this guy becomes COO minus. So then the charge is 0. So then this is one of the values that we should definitely consider. And then just the next value is pKa2, which is 9.6, and so this group becomes NH2, and so what happens? The charge is now -1, and so therefore we take two values that are closest to the pH at which zwitterion is formed. Therefore this is what we look into. The two values are 3.65 and 9.6, and the isoelectric point is 6.625. I would rather take a pen, it would be easier. So this is the pH, okay. So the value is 6.25, okay. So now there is again an MSQ on proteins versus free amino acids. So let us look at this question, and very soon we are going to end our class. So we just have 2 or 3 more questions left. So again, this is a multiple select question. So there are going to be more than one right answers. Okay, so let us look at this question. The titration of arginine residue present in a protein is difficult to carry out with NaOH, unlike individual arginine. Since, so what does the question say? It is saying that let's say this is a protein. Inside that protein, arginine is present. And if you compare it with this individual arginine residue that is present in a solution. This is a solution of only arginine, and this is a protein containing arginine. So the titration, NaOH titration, is difficult. Why is that so? Because here what happens, this arginine, the properties of this arginine is going to get affected by the properties of the neighboring amino acid. Now these are the different neighboring amino acids that are present, and we saw very recently that we need higher pH to neutralize, we need higher pH so that zwitterion can be made from histidine when it is present in a protein containing too many H plus charges surrounding it, right? So that is what we learned very recently in the last question. But then in case of this question, they are asking us why the titration of this particular arginine is more difficult compared to this free arginine. So the the things that they have given us, the options, these are protein forms complex with NaOH. Multiple amino acids may make the process complex. So this is definitely one of them, right? So multiple amino acids definitely make the process more complex. The structure of the protein may degrade at a higher pH. And then there is all of the above. So protein forms complex with NaOH. I don't think this is correct. So since this is not correct, I don't think it should be all of the above, okay? So let us look at the answer. The answer is this. The structure, okay, 1 minute. Multiple amino acids, okay, I think this is also the structure, this is also an answer. Since this is an MSQ, this is a multiple select question, and so B should also be correct. And they have also said that the structure of the protein may get degraded at higher pH. This is also one of the possibilities. So both of these answers are correct. So extreme pH can degrade a protein structure. In addition, it is not possible to estimate the pI of a particular amino acid in a protein due to multiple amino acids which can affect the measurement. Alright, so since this is an MSQ, we have both of these answers as correct. Right. Okay, they have already given it as correct, alright. So now, these are the different references of the books. These two are books. This is Leninger's Principle of Biochemistry. This is Voigt and Voigt's Biochemistry. So these are the two books that you can go through, and these are the links to different websites that you can refer to. Yeah, so these are the different websites that you can refer to for studying these concepts better. And thank you for your time. It was a very good learning experience for me, and I hope it was also useful for you. So we will meet again in the next week with another set of MCQs, MSQs, and a lot of different concepts of biochemistry to brush upon. Okay, if you have any questions, you can ask me. I'm stopping the presentation. Thank you so much. Thank you. Okay, we can end this meeting now if you don't have any question. Thank you so much. We will be stopping the recording now.